Chapter 56. Cancer, an overview. And our goal is to decode the machinery. We want to move past the fear and get right under the hood of the biology.
Exactly. The fear comes from the unknown. Right. But when you really look at the source material, cancer isn't. It isn't magic. It isn't a curse. It is a biological process. It's a mechanism. And once you start to understand the mechanism, how a normal cooperative cell just decides to go rogue, it becomes much less abstract. It becomes a problem of engineering.
A very, very difficult engineering problem, but engineering nonetheless. Now, before we get into all the genes and the mutations, I want to start with the context that the source lays out. It calls cancer the second most common cause of death globally, right behind heart disease. We're at about 9 million deaths a year.
It's a staggering number. It really is. But there is a nuance in the text that I think is really important to frame our entire conversation today. The rise in cancer rates is, in a very strange way, a paradox of our own success.
How so? What do you mean by that?
Well, think about it. For most of human history, you didn't live long enough to get cancer. You died of smallpox or tuberculosis or an infected cut or starvation. As we've engineered our way out of those early deaths, as sanitation and antibiotics extended our lifespan, we've basically just entered the window where cancer becomes a real possibility.
So, because we aren't dying at 30 anymore, we're sticking around long enough for our cells to just break down.
Precisely. Cancer is largely a disease of aging. It's the accumulation of damage over time. The source actually frames it as a battle against entropy, which I think is a fascinating way to put it. But let's define what we're actually fighting here. The medical term is neoplasm.
Which sounds like something out of a 1950s sci-fi movie. The attack of the neoplasm.
It does, doesn't it? But it literally just means new growth. And this is where people get tripped up. Not every neoplasm is cancer. If you have a mole on your arm, that is technically a neoplasm. If you have a uterine fibroid, that is also a neoplasm.
But those are benign.
Correct. And the distinction the text makes between benign and malignant is absolutely crucial. A benign tumor can grow, it can get big, it has what the source calls diminished growth control. But, and this is the key, it respects boundaries.
It stays in its lane.
It stays in its lane. It does not invade the tissue next door. A malignant tumor, which is what we call true cancer, is a rule breaker. It invades, it destroys the surrounding structures, and ultimately it gains the ability to metastasize, to pack its bags and travel to a different organ. That ability to invade and spread is the defining line.
The source has introduced this framework that I found really helpful. It's called the hallmarks of cancer. It's sort of a checklist of superpowers a cell has to acquire to become malignant.
It's the gold standard for understanding cancer biology. It was originally proposed by Hanahan and Weinberg, and it's brilliant. Think of it this way. A normal cell has a lot of safety checks built in. It only divides when it's told to. It kills itself if it's damaged. It stays where it's put. To become a cancer, a cell has to hack every single one of those safety checks.
It has to learn to grow without signals, ignore all the stop signs, avoid suicide, build its own blood supply. It's actually kind of terrifying how competent these cells become.
It is an evolutionary feat. It really is. And that's really the theme of this deep dive, evolution happening inside the body. But let's start at the very, very root of it, the software code, the genetics.
Right. Because fundamentally, the sources all agree cancer is a genetic disease.
It is. But not necessarily genetic in the sense that you inherited it from your parents, though that can happen, of course. It's genetic in that it is caused by damage to your DNA. The text calls this non-lethal genetic damage.
I paused on that phrase, non-lethal.
Uh-huh.
Why is that distinction so important?
Well, think about it. If you get hit by a massive dose of radiation that scrambles your DNA so badly that the cell just can't function, what happens?
The cell died.
The cell dies, and a dead cell cannot form a tumor. For cancer to start, the damage has to be in this, Goldilocks zone. It has to be bad enough to break the control systems, but not so bad that the cell can't survive and replicate. It has to survive the mutation.
And once that one cell survives the mutation, we get into this concept of clonal origin.
This is fascinating. Despite how messy and diverse a large tumor looks under a microscope, the evidence suggests that almost all tumors start from a single precursor cell. One bad actor that acquired the first crucial mutation and just started cloning itself.
So every cancer is basically a clone army descended from one single mistake.
Essentially, yes. Now, what actually breaks? The text divides the critical genes into two main categories. I always use the car analogy here because it's just so perfect for visualizing this.
The gas pedal and the brakes.
Exactly. To drive a car or to drive the cell cycle, which is the process of cell division. You need a mechanism to go, and you need a mechanism to stop. The go genes are called proto-oncogenes.
Proto meaning original or before.
Proto meaning the normal, healthy version. You need these genes. They code for things like growth factors, receptors, the things that tell your liver to regenerate after an injury or your skin to heal a cut. They are the gas pedal.
But in cancer, they mutate.
And they become oncogenes. The proto part drops off. Now you have a gas pedal that is just stuck to the floor.
The text calls this a gain-of-function mutation, which makes sense. You've gained a drive that shouldn't be there.
And because it's a gain of function, you're adding a proactive force, you usually only need one copy of the gene to be mutated. Remember, we have 2 copies of every gene, one from mom, one from dad. If just one of your gas pedals is stuck down, the car is going to accelerate, even if the other pedal is working perfectly fine. It's a dominant effect.
The source outlines a few ways this gas pedal gets stuck, and I want to drill down into these because the mechanics are just wild. Let's talk about the RAS gene. The text mentions point mutation.
Oh, RAS is a classic example. It's one of the most common mutations in human cancer. We see it in pancreatic, colorectal, lung cancers all the time. Normally, the RAS protein acts like a momentary switch. It sits right inside the cell membrane. When a growth signal hits the cell from the outside, RAS flips to on, binds to a molecule called GTP. It sends the signal grow, and this is the crucial part, it immediately cuts that GTP and turns itself off.
It has a built-in timer.
Exactly. It has what's called intrinsic GT-pace activity. It resets itself. But a point mutation, changing just a single letter in the DNA code, can destroy that timer mechanism.
So the spring in the switch breaks.
The spring breaks, RAS flips on, but it can't flip off. It is trapped in the active state. So the nucleus is getting this constant screaming message, divide, 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 and it's all coming from one tiny letter change in the DNA.
That is terrifyingly fragile.
Okay.
Another mechanism is chromosomal translocation. This one felt more like a construction accident.
It's a relocation error. That's a great way to put it. You're moving a gene to a neighborhood where it just doesn't belong. The textbook example here is Burkitt lymphoma.
This involves the MYC gene, right? I've seen that one come up a lot.
Yes, MYC. MYC is a master regulator. It's a transcription factor that turns on a whole host of other growth genes. Normally, MYC is on chromosome 8, and it's very tightly regulated. It only speaks when it's spoken to.
Okay, so it's a quiet neighbor.
A very quiet neighbor. But in Burkitt lymphoma, a piece of chromosome 8 breaks off and swaps places with a piece of chromosome 14.
And what's happening on chromosome 14 that's so important?
That's the home of the immunoglobulin heavy chain genes.
The antibody genes.
The antibody genes, yes. Now think about a B cell, which is the cell type that becomes cancerous in lymphoma. What is a B cell's job? It is an antibody factory. It is designed to pump out antibodies 204/7. So that genetic region on chromosome 14 is always open for business. It has incredibly powerful promoters driving gene expression.
So you take MYC, which is supposed to be quiet, and you drop it right next to the antibody factory's loudspeaker.
That's it exactly. You put the GROW gene under the control of the always-on promoter. The cell starts churning out MYC protein like it's churning out antibodies, and the cell is just flooded with growth signals.
It's a location-based disaster.
It is. So those are the oncogenes, the stuck gas pedals. But the car shouldn't crash just yet, because we have brakes.
The tumor suppressor genes.
These are the genes whose entire job is to say no. They stop the cell cycle, they fix damaged DNA, they initiate cell death if things are too far gone.
And unlike the oncogenes, these have a loss-of-function effect.
Right. If you cut the brake lines on your front left wheel, the car might still stop because the other 3 brakes are still working. In genetics, tumor suppressors are usually recessive. You need to knock out both copies, both alleles, to completely lose control.
This brings us to a really famous concept in the text, the 2-hit hypothesis.
Alfred Knudsen. This is a foundational idea in cancer genetics. He came up with it by studying a rare eye cancer in children called retinoblastoma. He noticed something strange. There were 2 groups of kids getting this cancer. Some got it as babies, and often in both eyes. Others got it a bit later in childhood, and usually just in one eye.
Why the difference?
He realized the babies were born with the first hit. They had inherited one broken copy of the RB gene from a parent. So literally every cell in their retina was already sitting there with one break line cut. They just needed one random mutation, the second hit to knock out the other copy, and boom, you get cancer.
Whereas the older kids,
they started with 2 good copies, so they had to be incredibly unlucky. They had to get a spontaneous mutation in the RB gene in one single cell, and then later on get a second spontaneous mutation in that exact same cell's other copy of the gene. Statistically, that just takes much longer to happen.
That explains the age difference perfectly. Now, we can't talk about tumor suppressors without talking about the big one, the guardian of the genome.
P53.
It sounds like a droid name from Star Wars, but it's arguably the most important single protein in cancer biology. The text says it's mutated in something like 50% of all human cancers.
Roughly 50%, yes. And in the cancers where it isn't directly mutated, its pathway is often deactivated by other means. You almost have to get rid of P53 to make a successful cancer.
What makes it so special? Why is it the guardian?
Because P53 is the central decision maker. It's a stress sensor, it floats around in the nucleus, and its levels rise whenever there is cellular stress, things like DNA damage, lack of oxygen, nucleotide deprivation. When P53 senses that damage, it acts as a transcription factor and it makes a call.
It has two options, right?
Two main options. Option one is stop. It triggers other proteins that halt the cell cycle. It puts up a red light. This gives the cell time to call in the repair crew to fix the DNA.
And that's sensible. Pause and repair.
But if the repair crew comes back and says, boss, this is too much, the DNA is shredded, we can't fix this, then P53 goes to option 2,
The nuclear option.
apoptosis, programmed cell death. P53 basically tells the cell, you are a danger to the organism, you need to die for the greater good, and it initiates the suicide sequence.
So if you lose P53,
You lose the stop sign and you lose the eject button. You have a cell with damaged DNA that just keeps on dividing. And because it has damaged DNA, the daughter cells have even more mutations. And they divide. It creates the snowball effect of genomic instability.
the source makes a distinction between gatekeepers like P53 and RB and caretakers. Can we clarify that?
Yeah, that's a useful distinction. Think of gatekeepers as the security guards who physically open and close the doors to cell division. They directly control the cycle. Caretakers are more like the maintenance staff. These are the DNA repair genes like BRCA1 and BRCA2 that you hear about with breast cancer. Their job is to constantly patrol the DNA and fix typos. If the maintenance staff goes on strike because of a mutation, the building starts to fall apart. Mutations accumulate all over the place, and eventually one of those mutations is going to hit a security guard. So loss of caretakers accelerates the mutation rate of the gatekeepers.
That makes perfect sense. So we've got the gas stuck, the brakes cut, and the repair crew is gone. But the text emphasizes that this doesn't happen overnight. It's a multi-step progression.
This is absolutely critical. You don't just wake up with cancer because of one zap of radiation. It's an evolutionary timeline playing out inside your body. The text uses the colorectal cancer model to explain this because we understand it so well, we can actually watch it happen.
Let's walk through that story.
Yeah.
Because the colon actually gives us a physical timeline we can see during something like a colonoscopy.
Exactly. It starts with normal epithelium, the healthy lining of the gut. The first event is usually the loss of a gene called APC. APC is a tumor suppressor. When you lose it, the cells don't stop dividing when they crowd up against each other. They start to pile up.
And this forms a polyp.
A small benign polyp, or an adenoma, technically. If you go in for a colonoscopy and the doctor snips out a polyp, they're literally stopping the story right there at chapter 1.
But if you leave it there,
The cells in that polyp are now dividing faster than normal. That means more chances for error, more chances for another mutation. Eventually, one of those polyp cells gets a second hit, often the RAS mutation we just talked about. Now it's growing even faster, the polyp gets bigger. It becomes what's called a late adenoma.
but it's still not technically cancer.
Not technically malignant yet. It hasn't invaded the wall of the colon. But then, as that chaotic growth continues, you might lose a gene on chromosome 18. And then finally, the coup de grace. You lose P53. Once P53 is gone, the genomic instability explodes. The cell gains the ability to invade through the basement membrane and into the wall of the colon. Now, it is a carcinoma.
So it's a specific sequence of events. APC goes first, then RAS, then P53.
Generally, yes, that's the classic progression. It's estimated you need hits in about 5 or 6 distinct signaling pathways to make a solid tumor. This is why cancer is mostly a disease of the elderly. It takes decades to accumulate that specific unlucky poker hand of mutations all in a single cell line.
This leads to the idea of evolution in real time. I think we tend to view a tumor as just a lump of identical bad cells.
Which is a huge misconception. A tumor is a heterogeneous mess. It's a chaotic ecosystem. Because the cells are genetically unstable, they keep mutating as they grow, so you develop subclones. One group of cells inside the tumor might be really good at growing fast. Another group might have a mutation that makes them really good at surviving without much oxygen.
It's survival of the fittest, but happening inside an organ.
That's precisely what it is, and this is why chemotherapy can be so tricky. You might give a drug that kills 99% of the cells, the ones that are sensitive to that drug, but if just 1% of the cells have a random mutation that happens to make them resistant,
They survive.
they survive. And now that you've killed off all their competitors, they have all the space and all the resources to grow back. The tumor returns, but this time it's made up entirely of cells that are resistant to the drug you just used. We have essentially selected for the supercancer.
That's a really sobering thought. We're applying evolutionary pressure. Let's pivot to the external enemies. We know the genes break, but what breaks them? Section 3 in the source covers carcinogens.
We usually group these into 3 big buckets: radiation, chemicals, and viruses.
Let's start with radiation, sunlight. We all know that UV causes skin cancer, but how? Is it just burning the cell?
It's much more specific than a burn. UV radiation acts like a physical hammer on the DNA structure itself. Specifically, it causes something called pyrimidine dimers.
Unpack that for me.
Okay, so DNA is a ladder, right? The rungs are made of letters. T connects to A, C connects to G. UV light has just the right energy to make 2 adjacent T's on the same side of the ladder fuse together. They stick to each other instead of their partners across the way.
So it creates a kink in the ladder.
A literal physical kink. When the DNA copying machine, the polymerase, comes along and hits that kink, it derails. It doesn't know what to read, so it often puts in the wrong letter on the new strand. That's a typo. That's a mutation.
And ionizing radiation, like X-rays or gamma rays from nuclear sources, is even worse.
Oh, much more energetic. It doesn't just fuse letters, it can strip electrons right off of atoms. It creates free radicals or reactive oxygen species. These are like tiny pieces of shrapnel flying around inside the cell. They can snap the DNA backbone right in half, what we call double-strand breaks. Those are incredibly difficult for the cell to fix correctly.
Okay, moving to chemicals. There was a concept here that really surprised me, the idea of procarcinogens.
This is a classic case of the body's own defense mechanisms backfiring in a terrible way.
Explain that.
Well, many chemicals, like the polycyclic aromatic hydrocarbons you find in cigarette smoke or soot, aren't actually carcinogenic when you first inhale or ingest them. They are chemically inert. They are procarcinogens.
So they're harmless initially.
Initially, yes, until they hit your liver. The liver is our main detox center. It has a family of enzymes called cytochrome P450. Their job is to take foreign chemicals, oxidize them, make them more water-soluble so you can pee them out. It's usually a great system. But for certain molecules, the very chemical reaction that is supposed to detoxify them accidentally turns them into a highly reactive monster. This new molecule is called an electrophile.
An electron lover.
Yes, exactly. And what is DNA packed full of? Electrons. So this newly created ultimate carcinogen rushes out of the enzyme and attacks the DNA, binding to it, forming adducts and causing mutations.
So the liver is trying to wash the poison out, but in the process, it accidentally turns it into a grenade.
That is the perfect analogy. It is a biological betrayal.
Speaking of testing for these chemicals, the text details the Ames test. I love this story because it's such a brilliant piece of low-tech biological hacking. How do you test if a chemical causes cancer without injecting 1,000 rats and waiting 2 years for tumors to grow?
Bruce Ames figured this out back in the 70s. It was genius. He used bacteria, a specific strain of Salmonella, but he used a very specific broken strain. These bacteria had a mutation that meant they couldn't make their own histidine, which is an amino acid they absolutely need to live.
So if you put them on a petri dish with no histidine in the food, they die.
They starve. The plate stays clear. That's your baseline. Now, here's the hypothesis. If a chemical is a mutagen, if it scrambles DNA, it might just randomly scramble the bacteria's DNA back to the correct working version.
A reverse mutation. You're fixing it by breaking it more.
A reverse mutation. It's like hitting a broken TV and having it suddenly start working again. So, Ames would mix the chemical he was testing with the bacteria and spread them on the plate with no histidine. If he came back the next day and saw colonies of bacteria growing,
it meant the chemical must have mutated the DNA, fixed the original defect, and allowed those bacteria to grow.
Exactly. The more colonies you see, the more potent the mutagen is. But, and this is the truly genius part, Ames remembered the liver problem we just discussed. He knew that bacteria don't have livers. If he just dumped a procarcinogen on the plate, nothing would happen. He'd miss it.
So what did he do?
He went to the lab supply and got rat livers. He ground them up into an extract, essentially a liver smoothie that contained all those metabolic cytochrome P450 enzymes, and he just added a little bit of that to the petri dish.
Gross, but absolutely brilliant.
It simulated the human body in a petri dish. The liver extract activated the activated chemical mutated the bacteria, and the bacteria grew. The Ames test. It became the standard first-pass screen for carcinogens, and it's still used today.
That is just science at its best. Okay, third bucket, viruses. I think people are often shocked to learn that viruses can and do cause cancer.
It accounts for about 15% of all cancers worldwide, so it's a significant factor. The big player here, especially in the developed world, is HPV human papillomavirus.
The cause of cervical cancer.
And a growing number of head and neck cancers, too. The mechanism here is just diabolical. HPV is a DNA virus. When it infects a cell, it wants that cell to divide so the virus can make more copies of itself. But a normal, mature epithelial cell isn't dividing. It's just sitting there doing its job. It's resting.
So the virus has to force it.
It has to force it into the cell cycle. And to do that, HPV produces two proteins. You can think of them as hitmen. They're called E6 and E7. The E6 protein finds P53R guardian binds to it and targets it for destruction. The E7 protein finds RBR brake pedal binds to it and completely incapacitates it.
It takes out the two most important tumor suppressors we have.
Systematically. It cuts the brakes and shoots the security guard. With those two gone, the cell is forced into the cell cycle and the virus happily replicates. The cancer is almost just a long-term side effect of the virus's own life cycle.
Incredible. So we've covered the what and the why. Now I want to go even deeper inside the cell's operations. Section 4 in the source talks about signaling and epigenetics.
We mentioned the gas pedal getting stuck, but there's another way that cells can trick themselves into growing. It's called autocrine signaling.
What's that?
Normally, a cell is polite. It waits for a neighbor or a signal from the brain to send a growth factor molecule. It waits for permission to divide. Cancer cells, well, they stop waiting. They start manufacturing the growth factor themselves.
It's like talking to yourself in a mirror and agreeing with everything you say.
And then acting on it. Exactly. They produce the signal and they possess the receptor for that signal. It creates this dangerous, self-reinforcing loop of grow, grow, grow.
And then there's epigenetics. This is a field that has just completely exploded in recent years. It's the idea that you don't have to change the DNA letters themselves to change how the genes are expressed.
Think of DNA as the hardware and epigenetics as the software running on it. Or maybe a better analogy, DNA is the script for a play. Epigenetics is the director's notes in the margin, crossing out entire scenes or highlighting certain lines.
How does the cell cross out a scene?
The most common way is through methylation. By adding a small chemical tag, a methyl group, to the DNA strand, usually at the promoter region of a gene, the cell essentially puts a piece of molecular duct tape over that section. The reading machinery, the transcription factors can't bind to it. The gene is silenced.
So in cancer,
they often use this to their advantage. They will hypermethylate the promoters of tumor suppressor genes. The gene is there. The DNA sequence is perfect. It's not mutated, but it's taped over. It's silenced. The end result is the same as a mutation. You've lost the function of that break.
And then there's this idea of immortality. Normal cells aren't immortal. They have a built-in lifespan.
Right, that's right. It's called the Hayflick limit. A normal human cell can divide maybe 50, 60 times, and then it just stops. It enters a state called senescence. This is due to telomeres.
The caps on the ends of the chromosomes.
Exactly like the little plastic aglets on the ends of your shoelaces. Every time a cell divides, the DNA copying machinery can't quite reach the very end of the chromosome, so a little piece of the telomere gets chopped off with each division, the shoelace gets shorter and shorter.
And when the plastic tip is completely gone,
the shoelace phrase, the raw DNA ends are exposed. The cell senses this as catastrophic DNA damage, and it enters senescence. It retires. It stops dividing forever to prevent itself from becoming a danger.
But cancer cells find a way to bypass this.
They do. They reactivate an enzyme called telomerase. Telomerase is a very specialized enzyme. It's like a repair kit for telomeres. It adds new DNA back onto the ends of the chromosomes. It retips the shoelace after every division.
So the cell never realizes it's old.
It can divide infinitely. For all intents and purposes, it becomes immortal.
I have to ask this. If telomerase is the key to immortality for cancer cells, why don't we just create a drug that blocks it? Wouldn't that stop all cancer in its tracks?
It's a huge area of drug development, and there are drugs in trials, but there's a big catch. We have normal cells in our body that need to be immortal, or at least highly replicative. Your stem cells, the cells in your bone marrow making your blood, the cells lining your gut, they all express telomerase too. If you block it systemically, you could crash the patient's immune system and their digestive tract.
The classic problem.
Ugh.
How do you kill the monster without killing the host?
Specificity is always the problem in cancer therapy.
Let's move to Section 5, death. We talked about P53 triggering suicide, but how does a cell actually die? The source makes it clear there are different ways.
There's a good way and a bad way for a cell to die. The good, clean way is apoptosis.
Programmed cell death.
It's very tidy, very organized. The cell shrinks, the chromatin in the nucleus condenses, the cell membrane blebs, and it breaks up into these neat little packages called apoptotic bodies. And then the neighboring cells, or immune cells called macrophages, come by and just eat them. It leaves no trace, no inflammation.
It's like taking down a building brick by brick, very carefully.
That's a great way to put it. And the central machinery here involves the mitochondria. We usually think of mitochondria as the powerhouse of the cell, making energy. But they are also little poison capsules. They're filled with a protein called cytochrome C.
Which is safe as long as it stays inside the mitochondria.
Right. But if the cell gets the signal to die via what's called the intrinsic pathway, it pokes holes in its own mitochondria. Cytochrome C leaks out into the cytoplasm. That leak is the trigger. It activates a cascade of enzymes called caspases.
Executioners.
They are the executioners, yes. They literally digest the proteins and the DNA of the cell from the inside out. Now, cancer cells defend against this by overproducing a protein called BCL2. BCL2 stands guard at the mitochondrial membrane and essentially plugs the holes. It keeps the cytochrome C locked inside so the cell can't die even if it gets the signal.
So that's the clean death. What's the bad death?
Necrosis.
Yeah.
This is accidental death. Say the tumor grows so fast that it outstrips its blood supply. The cells in the middle starve of oxygen and nutrients. They don't shrink, they swell up and they burst.
Like a balloon popping.
And when they burst, they just dump all their toxic contents, all their enzymes and proteins into the surrounding tissue. This causes massive inflammation. All the local immune cells rush in.
You'd think that would be bad for the tumor. And
Right.
immune response.
You would think. But the immune cells that rush in macrophages, neutrophils, they arrive to clean up the mess and to heal the wound. And to do that, what do they secrete? They secrete growth factors and enzymes to help rebuild the tissue.
And the surviving cancer cells just on the edge of that necrotic zone say, thank you very much.
Exactly. The tumor hijacks the body's own wound healing response to fuel its own growth. The inflammation acts like fertilizer. That's why we know that chronic inflammation, like in ulcerative colitis or chronic hepatitis, is such a strong risk factor for developing cancer in those organs.
That is a cruel twist. The body's attempt to heal actually helps the disease.
It's a recurring theme in cancer biology. The disease is a master of subversion.
Speaking of fuel, let's talk about metabolism. Section 6, the Warburg effect. This part always confuses me because it seems to violate all common sense.
It confused Otto Warburg back in the 1920s, too, when he discovered it. He noticed that cancer cells consume enormous amounts of glucose sugar. But instead of burning it efficiently in the mitochondria using oxygen, which is called oxidative phosphorylation, they ferment it into lactic acid. They do this even when there's plenty of oxygen available.
It's called aerobic glycolysis. But isn't that incredibly inefficient? I remember from high school biology, burning glucose in the mitochondria gives you like 36 ATP molecules. Glycolysis only gives you 2. It's like getting 2 miles per gallon when your car is capable of getting 36. Why would a rapidly growing cell choose the inefficient path?
That is the paradox. But you have to think about what a cancer cell really needs. Does it just need energy in the form of ATP? No. It needs to double its mass every few days. It needs to build a whole new copy of itself from scratch.
It needs building blocks.
It needs building blocks, exactly. It needs carbon. If you burn glucose all the way down in the mitochondria, you turn it into CO2 and you just breathe it out. All that precious carbon is gone. By stopping at glycolysis, the cancer cell keeps the carbon chains from the glucose intact. It can then shunt them off into side pathways to build nucleotides for new DNA, lipids for new membranes, and amino acids for new proteins.
So it's deliberately trading energy efficiency for biomass. It's hoarding the Lego bricks instead of burning them for heat.
That is the perfect analogy. And this metabolic quirk is incredibly useful clinically. Because they are so inefficient at making energy, they have to consume way more glucose than normal cells just to
They
survive.
have a massive sweet tooth.
A massive one. And this is precisely how PT scans work. We inject the patient with a form of radioactive glucose. The tumor cells, being so greedy for sugar, suck it up much faster than the surrounding healthy tissue. So when we take the scan, the tumor lights up like a Christmas light bulb.
That's fascinating. But there's a byproduct of all this fermentation, right? Lactic acid.
Yes. They are constantly pumping out lactic acid. This makes the entire tumor microenvironment very acidic. This is actually toxic to normal cells, and it has the added benefit for the tumor of shutting down the immune cells that are trying to attack it. It creates a protective shield of acid.
This brings us to the microenvironment. A tumor isn't just a ball of cancer cells sitting in isolation.
No, not at all. It's a rogue organ. If you were to look at a slice of a tumor under a microscope, a huge percentage of the cells you see are not actually cancer cells. They are corrupted fibroblasts, immune cells, blood vessel cells.
But they're all working for the enemy.
The tumor corrupts them. It tricks local fibroblasts into building a stiff scaffold of scar tissue around it, which is called desmoplasia. This protects the tumor. It tricks macrophages, which should be eating the cancer cells, into becoming what we call M2 polarized macrophages. These actually suppress the immune system and secrete factors that help the tumor grow.
And it builds its own plumbing, the process of angiogenesis.
A tumor can only grow to about 1 or 2 millimeters in diameter, about the size of a pinhead, before it runs out of oxygen. Diffusion can only go so far. It hits a physical wall. To get any bigger, it needs its own blood vessels.
That calls for them.
It starts secreting a molecule called VEGF, vascular endothelial growth factor. It's a chemical signal that basically screams, "Build me a highway!" And the local, healthy blood vessels respond by sprouting new branches and growing right into the tumor. But because they are built in such a rush under these chaotic signals, the new vessels are leaky, twisted, and disorganized.
Which brings us to the most dangerous phase of all, Section 7, metastasis. The source is very, very clear on this. Something like 85 to 90% of cancer deaths are due to metastasis, not the primary tumor itself.
That's right. If a cancer stayed put, we could usually just cut it out or radiate it. Metastasis is what turns a local problem into a systemic and often fatal disease, but it is an incredibly difficult and inefficient process for a cell to pull off. We call it the invasion cascade.
Let's walk through the steps of that cascade. Step 1, detachment.
Normal epithelial cells are glued tightly together by a molecule called E-cadherin. It acts like molecular Velcro. One of the first things invasive cancer cells do is they downregulate E-cadherin. They lose the Velcro, they let go of their neighbors and become individual.
Step 2, movement.
This is a wild transformation. The cell undergoes what's called EMT, an epithelial to mesenchymal transition. An epithelial cell is like a brick in a wall. It's rigid, stationary. A mesenchymal cell is more like a slug. It's soft, mobile. The cancer cell literally changes its shape and its internal skeleton to become a crawler.
Step 3, invasion.
Now that it can move, it has to chew its way through the basement membrane, the floor that its whole sheet of cells was sitting on. It secretes enzymes called MMPS matrix metalloproteinases. You can think of them as molecular machetes, just cutting through the jungle of the connective tissue.
Step 4, intravasation, getting into the bloodstream.
It crawls into one of those leaky, poorly formed blood vessels that it induced. Now it's in the bloodstream. And this is an incredibly hostile environment. The sheer force of the blood flow can rip the cell apart, and the immune system is constantly patrolling for anything that looks out of place.
How do they survive that journey?
They use disguise. Some circulating tumor cells attract platelets, the normal clotting factors in your blood, and they coat themselves in a little jacket of platelets. They essentially wear a cloak of normal blood components to hide from the immune system and to cushion themselves against the turbulence.
Step 5, extravasation, getting out of the blood vessel.
It has to get stuck somewhere. It tumbles through the circulation until it gets lodged in a tiny capillary at a distant site, say in the lung or the liver or the brain. Then it has to actively squeeze out of that vessel back into the tissue.
And finally, the last and hardest step. Step 6, colonization.
This is the hardest step by far. Just because you land in the lung doesn't mean you can grow there. The environment is completely foreign. The soil has to be right for the seed. This is the seed and soil hypothesis, which was proposed by an English surgeon named Stephen Paget way back in 1889.
Certain cancers prefer certain organs?
Exactly. Breast cancer loves to go to bone. Colon cancer very often goes to the liver. The cancer cell has to figure out how to survive and thrive in this totally new foreign environment. The vast majority of them fail. They land and they either die or they go into a dormant state for years. But if one single clone figures it out, that's how you get a metastatic tumor.
It's a marathon of incredibly difficult hurdles. It's actually amazing that any cell makes it through it all.
It is statistically very, very unlikely. But when you have a primary tumor shedding millions of cells into the bloodstream every single day, eventually one of them gets lucky.
Let's pivot to the hope. We understand this machinery better than we ever have before. How are we using this knowledge to treat people? Section 8 covers detection and treatment.
Let's start with detection. Biomarkers. These are substances that the tumor can shed into the blood that we can measure. Things like PSA for prostate cancer, CEA for colon cancer, AFP for liver cancer.
But the text notes that they aren't perfect for diagnosis.
PSA is a great example. It stands for prostate-specific antigen. The key word is prostate, not cancer. It is specific to the prostate, not to prostate cancer. If you have a benign enlargement of the prostate or even just an infection, your PSA can go up. So it leads to a lot of false alarms and unnecessary biopsies.
So they are better for monitoring the disease after it's been diagnosed.
Exactly. That's where they really shine. If a man has his prostate removed for cancer, his PSA should drop to 0. If it starts to slowly creep up a year later, chemically, the cancer has returned somewhere in the body long before you could ever see it on a scan.
And the future of this is liquid biopsies.
This is incredibly exciting. We now have the technology to sequence the tiny fragments of DNA that are just floating around in your blood. We can tell the difference between your normal DNA and the mutated DNA that was shed from a tumor. The sensitivity is getting good enough that we could potentially catch cancers much, much earlier than we can now.
Now let's talk treatment. We're moving away from the slash and burn era, as it's sometimes called.
Right. Traditional chemotherapy is cytotoxic. It kills anything that divides fast. That includes cancer cells, yes, but it also includes your hair follicles, which is why you get hair loss, and the lining of your gut, which is why you get nausea. It's carpet bombing.
The new era is precision oncology, or targeted therapy.
This is where understanding all that genetic machinery finally pays off. We find the specific broken gear in that particular cancer, and we design a drug to jam just that one gear. The poster child for this is Gleevec, or imatinib.
Tell that story, it's a great one.
So, a cancer called chronic myeloid leukemia, CML. We discovered it's caused by a very specific chromosomal translocation, the Philadelphia chromosome. This translocation creates a mutant fusion protein called BCR-ABL. This protein is a type of enzyme called a tyrosine kinase, and it is stuck permanently in the on position.
That gas pedal again.
A very specific gas pedal. So scientists designed a small molecule Gleevec that fits perfectly into the active pocket of that BCR-ABL protein and just sits there. It blocks it from working.
And the result?
CML used to be a death sentence within a few years. Now, patients take a pill once a day, and they live near normal lifespans. It turned a fatal cancer into a chronic, manageable condition, almost like diabetes.
That is the dream for all cancers. And then there's immunotherapy. This feels like the biggest breakthrough in the last decade, maybe longer.
It is. It's a complete paradigm shift. For a long, long time, we wondered, why doesn't the immune system just kill the cancer? The immune system is brilliant at killing virus-infected cells and bacteria. Why does it ignore this giant tumor?
It turns out it's not ignoring it. It's being actively deactivated.
Correct. Your T cells, which are the soldiers of the immune system, they rush into the tumor, ready to attack. But the tumor cells express a protein on their surface, often one called PD-L1. This PD-L1 protein binds to a receptor on the T cell called PD-1.
It's like a secret handshake.
It is a secret handshake that says, I am one of you. I'm a normal cell. Do not attack me. It's an immune checkpoint. It's a normal safety mechanism to prevent autoimmunity. But the tumor abuses this checkpoint to put the T cells to sleep right at the scene of the crime.
So the drugs.
Checkpoint inhibitors, drugs like Keytruda or Opdivo, they are antibodies that physically block that handshake. They put a piece of tape over either the PD-L1 on the tumor or the PD-1 on the T cell.
So the handshake fails.
And the T cell wakes up, it looks at the cancer cell and realizes, wait a minute, you're a mutant, you're not supposed to be here. And it unleashes hell on the tumor. When this works, the results can be miraculous. We've seen patients with advanced melanoma, a disease that used to be untreatable, have their tumors simply melt away.
But it doesn't work for everyone.
No, unfortunately not. Some tumors are cold. They don't attract T cells in the first place, so there are no T cells there to wake up. And you have to remember, when you take the brakes off the entire immune system, you can get autoimmune side effects. The T cells might start attacking your healthy liver or lungs, too.
Finally, let's touch on prevention. The text says a huge percentage of cancers are, in fact, preventable.
The basics matter more than people think. Tobacco cessation is the single biggest lever we could possibly pull to reduce cancer deaths. But also obesity. We now know that adipose tissue creates a state of chronic inflammation and releases hormones that can drive cancer growth. And vaccines. The HPV vaccine is literally a cancer prevention shot. We could effectively eradicate cervical cancer if we had widespread vaccination.
We've covered a massive amount of ground today, from that first single DNA typo all the way to metastatic spread across the body. If we have to summarize the mission of this deep dive here.
I think it's that cancer is a disease of the genome, but it's a disease of us. It uses our own machinery against us. It uses our growth signals, our blood vessels, our wound healing response. It is a corruption of our own biology.
Which leads to that final provocative thought from the source. It mentions that cancer is a disease of somatic evolution.
Think about what that means. We are multicellular organisms. We rely on the process of cell division to live, to grow, to heal injuries. But cell division is a mechanical process. It has a tiny but non-zero error rate. Entropy itself guarantees that over trillions of divisions, mistakes will happen.
So cancer isn't really an external invader like a virus.
No. In many ways, it is the statistical price we pay for being complex, multicellular life forms that live for a long time. As we age, as we accumulate more and more cell divisions, the probability of that one perfect storm of mutations happening in one cell just keeps rising. It is, in a very real sense, a battle against the fundamental mechanics of life itself.
That sounds incredibly grim, but also oddly clarifying.
It shifts the goal, I think. We might not cure cancer in the sense of eliminating it from the human experience forever. That might not be possible. But with precision medicine, with immunotherapy, we can turn it from a terminal event into a manageable one. We can outsmart its evolution.
We can jam the gears.
We can jam the gears one by one.
That feels like the right place to land. The complexity is the enemy, but our ever-deepening understanding of that complexity is our greatest weapon. Thank you for diving deep with us today.
It was a pleasure.
We'll get you on the next one.